But the first example I wanted to show you today is a project led by some of our customers in ETH Zurich, where they create a bioprinted perfusable skeletal muscle tissue. So they have this model that you see on the top left, where they create a little stretch of muscle tissue with the blue material that you can see, then interspaced with pleuronics that they can then remove to create the vascularization, and some handles on each side to be able to attach this this model. And so you'll see why very shortly. So they created different versions of this construct with and without channels, with and without anchors, and applying or not some tension to this construct. And very quickly, basically what they do is that they print these models and then let them proliferate for 4 days so the cells can sort of settle in there, proliferate and expand. And after that, once the population is settled or the cells are, comfortable again, they're placed in this specific platform where they are attached around pillars. So then you can also apply some tension if required. And so in there, they can be placed in there for about 11 days. So in total, it's 2 weeks. And by the end of these 2 weeks, you have like a fully differentiated muscle construct. And if you look closely on the model, so you have to recreate this interface between, for example, muscle and tendon, you have created this interwoven layers so that you can also increase the attachment between the 2 layers so you don't get the construct separating after a while. So here you can see that actually after 2 weeks, the whole construct was still staying together, and even with the tension applied, it was still staying cohesive. So that's a very important point. What we can see here as well is the role of the vascular or the channels that they created inside, which is both essential for to allow the diffusion of drugs or materials through the construct. So you can see the difference in the time frame between sample with channels on the left and without channels on the right. But this is very important for oxygen diffusion. Because if you don't have, in larger constructs, you often have an oxygen gradient, and eventually you have hypoxia and necrotic cores in center of bigger constructs because they don't have access to oxygen. And with adding these channels, you can actually really decrease hypoxia within the constructs, and this really allows you to make much bigger models, which is quite important if we think about implantation also later in the future. And finally, the ability to create this tension on the construct really also helps the muscle cells to mature in a different, in a unique direction. So then in the end, the product that you get is a very much uniaxial growth of the muscle cells and a system that is very resembling actual muscle. And so once they had their model, optimized and set up, and they had confirmed that it was actually a proper muscle tissue that they had recreated, then they tested different options to sort of mimic an intramuscular injection of drugs within this construct and seeing how it would diffuse inside the muscle itself, and also through these channels that kind of reproduce blood vessels as well. So this is a very complex or quite complex model that can really not necessarily to be implanted in patients yet, but also represent a very valuable option for in vitro testing and have proper answers for experiments.